Hi, Miss Patterson here. Today we're going to talk about plasma membranes. This is part of AP Biology, Unit 2. So when we talk about plasma membranes, we need to go back and talk about macromolecules again. Macromolecules is something we're going to be talking about a lot. So if you need to go back and review, go back and look at my lipids video. So phospholipids are a lipid, and these make up the plasma membrane of cells. The parts that make up a phospholipid is I have what's called a hydrophilic head. And hydrophilic means it's water loving. It likes to be around water. It's going to attract water. So this phospholipid head is going to be at the top. I have a phosphate group, and if you remember our functional groups, phosphate is polar. It likes to bring in water because it's charged. These oxygens really greedy for electrons, so they're trying to get all this water. So this end here is going to really like water. I have a glycerol down here at the bottom, and then underneath that I have what are called hydrophobic tails. And hydrophobic tails are water-fearing. They don't like water. And if you remember in my video, we talked about these little peaks and valleys, and this represents hydrocarbons. And hydrocarbons are carbons, so each of these little points is a carbon, and off these carbons are a bunch of hydrogens. And these are very nonpolar. They have similar electronegative pull, so their electrons get shared equally. So there is no charge. There's no pull for water to want to come to it. So these stay nonpolar, which means they're hydrophobic. They don't like to be around water. Some will have kinks. If they have a kink, it means they have a double bonded carbon somewhere, and this becomes unsaturated. If I don't have any double bonds, it's going to be a straight line, and these are saturated fats. So these tails can have saturated and unsaturated fatty acid tails. So I have hydrophobic tails, and when I line them up, they're going to line up just like this. My hydrophilic heads are going to be around an aqueous solution. So this could be the outside of the cell, the exterior part of the cell. This here could be the interior part of the cell. And this is going to have some type of water environment. But this here is pretty watertight. It's not going to want water to be in here. So they're naturally going to be attracted to each other. This is what's going to be called a phospholipid bilayer. Bi means two. So a phospholipid group here, and it's a bilayer. We also call this amphiphatic. Amphiphatic means that I have material that is both polar and nonpolar, hydrophilic and hydrophobic. So that kind of is like both worlds living together in one molecule. I have saturated and unsaturated fats. So here I can see this molecule. These are those little fatty acid tails. And when I have them like this, I have these double bonds. This is going to be unsaturated. When I have any bonds, I can have one, I can have multiple, and this will make an unsaturated fatty acid tail. And they're going to be kinked. They're going to be bent out like so. The ones that are saturated, they're saturated with hydrogens, and there's hydrogens all over the place. And these are going to be very straight. And so these are saturated fatty acid tails. So when we look at these plasma membranes, I can have two types. Ones that have unsaturated fatty acid tails are going to be very fluid. These are going to push, and they're going to shove against the phospholipids around it, and it's going to push, and so it's going to make it flow more. This membrane is going to be more less viscous. Now the ones that are saturated, where I have all of these really long tight tails, they're going to pack up nice and tight. So when I have saturated fats, they're more of a solid, like butter. That's going to be more solid. All those saturated tails are going to be nice, packed tight together. But something like olive oil it's a fat, but it's going to be more fluid. These tails are keeping these phospholipids from coming in tight together. So we see that in membranes. So ones that are going to be more stiff, more viscous are going to be more saturated, and the ones that are more fluid, more able to move, are going to be unsaturated. When I have a plasma membrane, I have an internal and external environment, and this is going to protect the inside of the cell. This is very selective. So here I see my hydrophilic heads, I see my hydrophobic tails, my phospholipid bilayer, and this is going to help separate. This is going to allow material through and be very selective. I have my hydrophilic side, my hydrophobic area here. Remember, this is going to be just almost watertight, okay? And then I have what's called a fluid mosaic model. And this is an example of that. It's fluid, which means it's going to move. So all of these phospholipid heads are going to shift, and all these proteins that are in here are going to move. So this is constantly moving, shifting. This could be here, and then it could move and be down here, and this could move to the back. And so this is constantly always going someplace. That's always in motion. They're held together by those hydrophobic interactions. Remember, these hydrophobic tails don't want to be around water, so they're going to stick tight to each other. They're not going to want to try to pull away, so they're going to stay together. Again, it's going to move, it's going to be very flexible, and it's composed of many different molecules. So here I have my lipids, I've got my phospholipids, I have proteins that are going to be in here, I have carbohydrates, I've got some more types of lipids, these steroid cholesterol pieces are going to be in here. So I have a lot of other things besides just phospholipids living in this plasma membrane. Temperature can affect a plasma membrane. If I have unsaturated hydrocarbon tails, they're going to help maintain fluid movement. It's going to allow stuff to move through. So this works well with animals that live in really cold environments. So you think about fish that live in the Arctic Circle. How can these fish live in some place so cold? Humans couldn't live there. So how can fish? Fish have unsaturated fats. And so those are these tails that kink out, and it prevents the cells from kind of like freezing. It's going to allow them to be in colder water. It's not going to allow these to pack real tight together. So these are going to benefit animals or fish that live in really cold environments. So those kinks are going to prevent those tails from packing really tight and turning it more into like a thicker viscous or more of like a solid. And the big takeaway from this is cholesterol. Cholesterol is going to help maintain fluidity at high and low temperatures. And you find cholesterol in animals. Okay, places where you're going to have some type of animal is going to have some type of cholesterol. And what that does, which is really interesting, is when the membrane gets hot. So remember, when things heat up, the molecules move and they start to shake and they start to spread apart. The cholesterol will grab those tails and those phospholipids and hold them and keep them together and keep them from getting pulled out too far, so they keep them inside. The vice versa, when it gets really cold, molecules slow down and they get close together and they start to compact. And then what happens is when cells get cold, that membrane starts to come in. And now you have that cholesterol pushing away and like not letting it come in and get real packed. So that cholesterol is pulling together the membranes when it's hot, keeping it from expanding out. And when it gets cold, it's keeping it from compressing in and it's pushing against it. So cholesterol, you need some cholesterol for your cells to be able to do that. So at high temps, it's going to pull on the tails to keep those phospholipids close. At low temps, it's going to pull to keep them from packing together. Your membranes have proteins. You have what are called integral proteins. These are embedded, which means they're going to go through that layer. They are also called a transmembrane protein. So trans means across. If you ever heard of transatlantic crossing or transatlantic flight, they're going across the Atlantic. So transmembrane is going across the membrane. Again, amphipathic. You guys remember what that means? It means that I am the best of both worlds. I'm hydrophilic and hydrophobic. And it has to be, because look, I'm in a hydrophilic area here. I'm with all these hydrophilic heads, and then I'm having to live with all these hydrophobic tails. So I have to be able to play in both playgrounds. So I have to be able to do both. Peripheral are on the outside. If you have peripheral vision, it's out here. So peripheral proteins are going to be on the outskirts of this membrane. They're going to stay on the outside. They're not embedded, which means they're not going to be all inside of it. They just kind of stay on the outside, and they're just loosely bonded to that surface. So these proteins are pretty integral. They're going to allow material to pass through. They're going to allow stuff to get modified. These are going to be very big on allowing material and being basically the gatekeepers that help to move stuff through the cell. Next, we have membrane carbohydrates. These are going to recognize pieces in the cell. You have glycolipids, and glycolipids are going to be attached to a lipid. So that phospholipid head here, you can kind of see right here, it's attached to a lipid. And these are going to help recognize pieces in the cell. Glycoproteins, these are going to be attached to carbs. So these are going to be carbs that are attached to proteins. And there's quite a few of those. And what these do is they recognize parts of your other cells. If you have a foreign invader comes in, let's say you have a bacteria that's come in and it makes you sick, your body has to be able to recognize that. So I tell my students, you just need to think of it like all of your cells, this is a picture of your face. This is your identification card. And when another cell comes up to one of your other cells, they show each other their identification cards. Hey, this is Mrs. Patterson. Hey, this is Mrs. Patterson too. They high five, all right, and they go off and do their thing. If one comes up and says, hey, I'm Miss Patterson, and somebody shows their card and they're like, whoa, we don't know who you are, you're leaving, and they're going to basically escort them out by destroying them, bringing in the immune system, and they're going to start to destroy that. This is kind of what happens a little bit when people get organ transplants. You are getting an organ from somebody else that have all of these markers. They have somebody else's ID cards, basically, on those organs. And now they're being introduced into your body, and so your body doesn't recognize this person, this group of cells. So they have to break down your immune system to try to keep that from being such a problem and your body attacking these new organs that you've gotten if you have to have an organ transplant. So there's my glycoproteins, my glycolipids. So those are pretty important for cell recognition. All right, so movement through the membrane, I can move material back and forth. If they are small, non-polar, or gases, I can go through this membrane. I don't need any type of protein transport system or anything like that. So if I have things that are hydrophobic, because remember, they're going to want to be inside here, and they're going to be hydrophobic, they can go through. If I have carbon dioxide, which is small, it's going to be non-polar, it's going to allow me to go through. And then oxygen gas. This one here gets people. They're like, hey, whoa, but like oxygen, super electronegative, super greedy for electrons. But oxygen gas is O2. When oxygen bonds with oxygen, it turns nonpolar because my electronegative charges are equal. So these electrons get shared equally. So when I have oxygen gas, it's actually nonpolar. That's why it's able to pop through that membrane. Now, if I have bigger ones and I say if it's large and charged, it can't go through. So large ones are going to be too big. They have to go through some type of transport protein. If they are going to be charged, which means they are going to have a positive or a negative charge. Remember, those are going to be polar. They're going to attract water molecules to it, and they're going to not want to go through this part right here. So I can have stuff go through this transport protein, but what are really good transport proteins are what are called aquaporins. What do you think this gets through? Water. So water will go through here. Remember, water is polar. It can't go through this on its own. It's not effective. You might be able to get like one water molecule in here and it's going to bounce around like crazy trying to get it out, and it's going to be hard, and it's not going to be efficient for the cell. But these aquaporins are going to allow tons of water to go through. I'm talking like they can do over 3 billion water molecules in one aquaporin per second. So you think one Mississippi, boom, 3 billion water molecules just went through one aquaporin. That's not to mention all the aquaporins that are in your cell. So it's really good at transferring material back and forth. Plant cells, the cell wall outside that covers it is going to be covering a plasma membrane. So when we talk about isotonic and hypotonic and hypertonic, we're going to talk about these cell walls. And in the inside of it, you can see this plasma membrane. So you have this tough cell wall, it's going to support, it's going to protect it. But right on the other side, you have this plasma membrane. And the plasma membrane can move and pull, and it's going to allow material in and out of the cell. So I'm not going to have cell walls in animal cells. They're only going to be in plants. And here you can see these bricks. Those are my cell walls. And then on the other side of that cell wall will be that plasma membrane. That's going to help prevent material from just going wherever it wants. It's going to be a gatekeeper allowing material in and out. It's also going to, cell wall is going to help with water intake. It's going to prevent water from being evaporated out too quick. The cell wall is made of cellulose, and cellulose, remember, is a type of carbohydrate. It's insoluble in water. If you watch my carbohydrate video, we have alpha glucose, beta glucose, and they're going to switch, so I have bonds going in different directions. That's what gives cellulose such great support. And inside this cell wall, they also have what are called plasmodesmata. And these are little tunnels or little doorways, I like to call them, that allow you to get into the cell. So I go through this cell wall, then my cell membrane is right there on the other side of that cell wall. That plasmodesmata is what's going to allow material to go from one cell to the other. They're like little tunnels that have a little bit of cytosol and like cytoplasm that will kind of go allow you to move material. So if I have to get something from this cell to this cell, I can go through this plasma membrane, and then I can go through here, and I can go, so I can travel through these walls. They're not just solid walls that I can't get through. Okay, that is it for, gosh, what was this one? Plasma membranes. All right, so if you guys have any questions, you guys let me know. We'll talk to you later. Bye.